So today we will talk about role of genetics in aging. So it's again very important, I believe, important discussion. Let's go. As always, I recommend you to download presentation as PDF file from the link in the description, and you will get access to the links and also to the source of information. Make your own research. Don't trust on my opinion because I am professional scientist, but I have a PhD in physics and mathematics, and only 5 years, last 5 years, I work in biology. So, and I'm not trained medical professional, and you are not supposed to follow my opinion. But yet, for me, at least for myself, it is. I consider what I do is right. Because at least I can think rationally and use scientific method for the research. Let's go. So the scientific study of heredity began with Gregor Mendel in mid-19th century. Before Mendel, ideas about inheritance were largely observational and theoretical. Mendel introduced controlled experiments and described clear principles of inheritance that founded experimental genetics. By the late 19th century, microscopic studies identified long spindle-shaped chromosomes in dividing cell nuclei as the carriers of hereditary material. The genetic material. The gene concept and the central role of DNA was solidified during the 40s and 50s. Okay, so this is what we know. The molecular basis of inheritance was transformed in 1953 when Watson and Crick described the double helix structure of DNA, explaining how genetic information is stored and copied. This discovery led to rapid development of tools and technique, and in particular DNA sequencing, recombinant DNA technology, and later high throughput genomics, which clarified gene function, gene expression, and protein biosynthesis. These advances enable large projects like Human Genome Project, and it founded in about $3 billion American by United States government. The Human Genome Project produced its first public draft in 2003. Since then, sequencing cost fell dramatically from $1 million to 2007 to only $600 today, so it's quite affordable, at least for some people, making personal genomes increasingly cheaper and common. Okay, so this is how it was developed, and now it is some genetic research can be done for only $100 American. So expectation for immediate clinical revolution after Human Genome Project were tempered by practical challenges. Translating genomic findings into therapies proven slower and more complex than hoped, and many DNA sequences initially labeled as junk have unknown functions or complex dependent roles. Some have inherited later functional discoveries as supporting non-standard views, such as intelligent design theory, which argues that some biological features are best explained by intelligent causation rather than solely by mutation and natural selection. Again, it was a very big step, but despite expectation, these genes didn't explain why we are humans, why animals are shaped like this, how new species appeared, etc. So it was very, very interesting dynamics in the science, in biology. So intelligent design theory contrasts with evolutionary theory, which explains diversity through mutation, selection, and other evolutionary forces related to genes. And debates continue, and gaps and unresolved questions in evolution explanation, for example, the rapid diversification of animal life during the Cambrian explosion, and it contradicts mainstream evolutionary biology and offers multiple hypotheses. So the research is continuous in this area. So it is again was very very special event. So it was actually mathematically demonstrated that the base of life on this planet and maybe elsewhere is intelligent design, not just mutation and evolution. Okay, so again, Cambrian explosion happened 530 million years ago, when a new species of unprecedented complexity appeared, and there is no explanation, evolutionary explanation to this event. And then even now, opinion dominates that humans were not evolved from primates, from monkey. So it was all designed. Okay, what else? What else is interesting? Consciousness is another unresolved scientific and philosophical problem, because again, previously humans were considered as bio-robots, and all brain and consciousness, it is just a product of the brain work, but again, it explains nothing. Schrödinger and others have speculated about deep links between physics and life, including possible roles for quantum phenomena, and contemporary proposals like quantum theory of consciousness are debated but not widely accepted. So the situation is such that, again, that physicists like Schrödinger and Werner Heisenberg were speculating or discussing that the base of all life and all universe is some consciousness field, quantum field. And then this quantum field is actually personal. It works as a person, and all what we have in this universe, both living objects and material objects, are all not only created but also supported by this consciousness field. Okay, alternative controversial ideas, for instance, Rupert Sheldrake morphic resonance, which invokes an external field carrying collective information across generation, also exist and are discussed alongside more conventional theory of the mind, such as Carl Jung, collective unconscious, etc. So it is like the experiences and memories of not only humans but also other animals. They contribute to this quantum field of consciousness, and they do not disappear, but they work, and they are shaping the other animals, and shaping the development of life, and development also of collective unconsciousness. So it is again very complicated questions, but it develops further, so we can expect some connection between Christianity and physics also already, and maybe connection between consciousness and life. So genetics has shaped many theories of aging, but again, yet this mechanistic idea that everything is defined by genes was dominating and is dominating till now. So then people, some materialistic scientists, they were thinking about aging due to genetic mutations of genes, or degradation of genes, etc. So some genetic variants are associated with longevity in humans, and the experiments in model organisms show that specific mutations can extend lifespan. And the somatic mutation theory proposed that accumulation of mutations in non-germ-like cells contributes to aging and age-related disease. But the degree to which somatic mutations drive aging in humans remain uncertain. Again, I don't consider this theory as true. It is, okay, but let's go further. You can read these publications at least. Increasingly, researchers emphasize that genes alone do not determine biology. Interactions among genes in the environment cannot be described only by genes. And also this cell context critical. Okay, let's go. So again, there are many theories of human aging, and it is gene mutation theory is one of them. Yeah, but also there are many others, and honestly, I don't. Okay, so it is most of them appeared not true. Maybe waste accumulation theory is closer to be true, and again, but it is not. It depends what is considered as waste. And I think it is waste. Even accumulation of waste, it contributes, of course, but it is not the primary cause of aging. Okay. Also, the oxidative damage. So if we consider oxidized proteins and oxidized lipids, and damage to DNA as waste accumulation of waste, yeah, we may think that it is again a part of aging, but again, it is not the primary cause. It is only secondary. Okay, I was talking about primary cause of aging as stiffening of the extracellular matrix. Okay, epigenetics. Epigenetics, again, very popular, a lot of activity, a lot of money go to this area. So it is like heritable changes in gene expression that do not alter DNA sequence, but they are defined as methylation rate of DNA and histone modification. It provides a mechanistic link between environment and gene activity. Epigenetic changes accumulate with age and have been proposed as contributors to aging, or also even measurements of aging. So, and measures derived from DNA methylation pattern, they called epigenetic clocks. They estimate, from this people estimate biological age, and most popular is Hoover's multi-tissue clock developed in 2013. And it is a commonly cited example. It is very, very popular. A lot of funds go to this Hoover epigenetic clock. And the epigenetic age, in principle, correlates to some degree with physiological markers like lung function, blood pressure, resting heart rate, and mortality risk. But measurements can vary by tissue, sampled clock algorithm, formation, in the immunity, and even time of the day, and other factors. So it is, I consider it is very unreliable measure of age and also expensive. So instead, I propose to use physiological biomarkers like lung function and blood pressure, resting heartbeat rate, etc. It's much better. Okay, but nevertheless, it exists. So it is both methylation of the DNA, so some part of some genes are marked, or they get this methylation group, but also the shape of histones. Histones are special proteins which participate in expression of this or that genes. So it is, yeah, it is quite complicated stuff, but environment is changing both. So it is again. So what else? Let's go to this epigenetic clock. So with epigenetic clock, what was the maximum predicted maximum lifespan and observed, versus observed lifespan. It is in logarithmic scale. So we see that humans live actually much longer than it is supposed to from this epigenetic clock. The humans live longer. Also, some whales live much longer, and also some like bats and mole rats live much longer. I cannot find it here. Okay, so maybe this. Okay, they live much longer than it is expected. Again, if we check these measurements, you see that it is measurements in various tissues, like DNA methylation rate and versus aging. It is very difficult to get some correlation, or to make some estimation. So you see the error can be very high, and also sometimes it goes in opposite direction. So with age, DNA methylation rate is reducing, so it is go in opposite direction. So it is, I believe that the use of this epigenetic clock is very much unreliable. It gives nothing from viewpoint of practical value. Instead, if we use pulse pressure is this difference between systolic and diastolic blood pressure, and this is much more reliable as estimation of the biological age of the organism. So it is, again, I was talking about this, you see, it is it depends on humans. Some humans aging slowly, some humans aging quicker, but in principle it is growth. It is very good marker to estimate biological age of the organism instead of this expensive methylation clock. So the commercialization of epigenetic aging measurements has become a part of a broader multi-billion-dollar anti-aging industry, offering supplements, procedures, and diagnostic. Again, people like the idea that genetics has potential to change aging, that manipulation with genes can help to live longer. My opinion, it is wrong. It is secondary. But yet again, it is multi-billion-dollar industry, and a lot of money go to this research and also to these measurements. And also, like some interventions are promoted to reverse biological age, include dietary supplement, in the transfusion of young plasma from donors, and stem cell therapies. They result after these interventions. The epigenetic aging of the person is estimated, and it gives some results like rejuvenation 20, 40 years, but then it is only temporary. It is in 2 months, in 2-3 months, these measurements go into normal. There is no reference to physiological markers, to your appearance, to muscle strength, to lung functions, etc. So reported benefits are, as I said, are often temporary. Many procedures require repeated application every few months, and cost range widely from hundreds to hundreds of thousands American dollars. But again, some wealthy people use it and rely on it, so they are spending millions American dollars per year on these measurements and on supplementation. For example, this calcium alpha-ketoglutarate acid The salt of alpha ketoglutaric acid is promoted as dietary supplement, which gives some fantastic rejuvenation by epigenetic agent, but it is no correspondence to physiological data. Okay, so this stem cell therapy, for example, a very good example. It can be very expensive, and it carries also risks such as immune reaction, inflammation, thrombosis, and potential tumor genesis. It is again very, it is cancer. Side effects can be cancer development, etc. All side effects depending on the source of the stem cells, delivery method, and the health of the patient. So, but again, it is, you see that stem cell industry is developing very quickly. It is 11.4% per year. So people making a lot of money, and it is very much promoted. But again, no, it's only temporary. People who make this stem cell therapy, they have some for a few months, some improvement in health and in feeling better, feeling excited. But then it goes after recovery. Next year they look even much older. I see such evidence from some enthusiasts who promote this stem cell therapy. Yeah, well, okay. Then again, you can see that such organization like Harvard University and other universities and medical professionals, they are actually not honest. So it's again, there is a scandal exposing more than $500 billion American fraud in stem cell research. So it is very dark area of science. So cutting-edge genetic approaches, including translator programming with Yamanaka factors and mRNA-based modalities, being explored for their potential to reverse cell age. Yamanaka factors can induce pluripotency or in cells also to return cells into stem cells. Also, RNA vaccine technology is developing, and especially a lot of money go to this vaccine technology, and we see the terrible consequences of this vaccine after COVID. So some of my fellow students got this vaccine and they passed away in a year or two after vaccination and boosting. So again, but this technology again, it's trillions of dollars go to this, trillions. And Harvard also promoting this Yamanaka factor, which are dangerous, but it is a lot of money go to this area. So while these avenues are promising, they are experimental and require safety and efficacy validation before clinical use. Yeah, so you see what was with this mRNA vaccine. Terrible results, terrible. But yet it is, nobody is, it was a fraud, but again, it was a crime actually. But what is, how Yamanaka factor works? The injection of some molecules or number of molecules to cells or tissues turns the somatic cells to pluripotent cells, and it is like it gives rejuvenation if it is measured by epigenetic clocks. Certainly it gives rejuvenation, but not improvement of the physiological factors. It also, not to mention huge risk in cancer development, etc. So beyond genetics and molecular therapies, lifestyle and physiological approaches have robust evidence for improving health span. lung function, blood pressure, heart rate, and measures of physical endurance are practical low-cost markers that correlate with long-term outcomes. So it's instead of this expensive method to measure biological age, which are practically no value, we can use very low-cost markers to estimate biological age, and with these markers we can estimate what method is helpful to rejuvenate your body or what is not, what diet is better, etc. So, for example, epigenetic age acceleration is associated with reduced lung function and improvement in some physiological markers. Again, there is some correlation between epigenetic age, biological age measured by epigenetic clock, but correlation is not exactly, it is again very expensive. What is improvement? It is useless to use them. Okay, yet again, it was measured that decreased diastolic blood pressure correlates with epigenetic age measurements, so it has been reported alongside with a reduction in measurement epigenetic age in small interventional studies, but causality and long-term benefit remain under study. Okay, spirometry is the most reliable the measure of morbidity and mortality and the risk of mortality, but it is not routinely used as a predictor in clinical practice. When I was young, spirometry was in every clinic. We were measuring lung capacity. It was so easy, so common. Now I go to some hospital, I don't see this spirometry. It is not available, and I cannot find some devices. I do these measurements myself, just with some plastic bottle, measure the maximum lung capacity, and if it is improving, so it's again, the maximum lung capacity, for example, correlates with biological age very, very much, so very reliably. But measurements are very cheap, yet, and also it takes time. It is not quickly changing, but if you see reduction in the maximum lung capacity, it is aging, and if it is improving, then it is rejuvenation, so it is easy to measure. So how this lung capacity, it is maximum lung volume, pardon, how it is changing normally in people. Different people have different maximum lung capacity, but yet for almost all people, they go down. You see, they go down, and but rate of this going down, it depends on your lifestyle or your diet. So some people die very quick because this lung capacity is quickly reducing. So again, I used to have lung capacity 6.3 liters, and now I have like 5.8, I don't know, 5.5, I already don't remember, but good sign that it is not dropping. It is not dropping. The lung capacity of people of my age already 3.3, 3.6 liters for my height, for my body. But so it is, my lung capacity is much better than normal adults, other adults. But again, it is not. These measurements you can be done, but you will not see the changes in your aging or rejuvenation quick. So the pulse pressure works much better. So okay, what else? Intermittent hypoxia, hyperpoxia training. So there are such methods of training, in high-intensity interval training and breath-holding exercise and free diving style training can stimulate physiological stressors that some studies link to reduce senescent cell burden, lower chronic inflammation, and improved cognitive function. However, more research is necessary to define safety and efficacy in humans. Again, I am a diver myself, and I use also this high-intensity interval training during cycling and in the morning, for example. I do it quite every day. And intermittent hypoxia, hyperpoxia is more complicated, but also can be useful. And these exercises are easy to do and easy to practice. They are very helpful to keep your lungs working well and to decelerate aging or even improve aging. So, okay, this is benefits of intermittent hypoxic training. As I said, different types of training can be done, but it is, again, it's very helpful to slow down aging or reverse aging. The microbiome, particularly the gut microbiota, contains orders of magnitude more genes than human genome and actively influences host physiology and markers of biological age, including epigenetic measurement. You see that people can measure your biological age, but it is defined, the deviation from to rejuvenation or accelerated aging. They are defined by your your microbes in the gut. If this microbiota is healthy and beneficial, you will the results will be younger, and if it is full of some bad bacteria, then it will be older, but it actually also works. So instead, again, mind that, again, these epigenetic measurements, they rely on genes in your gut. So how you, and the genes in your gut depends on your diet, and how you can correlate this epigenetic age to physiological age, it is, again, it is stupid. So the number of genes in microbiota in your colon up to 10 times more than in your body, and again this it is changing, it is you changing diet, it is changing. So mind that this all these genetic measurements are not reliable. Pathogenic gut microbes and their metabolites can promote inflammation and DNA hypermethylation, you see. So if you have gut microbiota is full of pathogenic microbes, then you will have accelerated aging. So it is, again, potentially, but yet again, there is correlation with this. Diet, prebiotics, exercise, and some antibiotic, use of some antibiotic in slow dose. They shape gut composition and function, making dietary modulation a powerful low-cost strategy to improve health span and to extend the longevity. It is, again, easy to do. Your diet, prebiotics, exercise, some medications and some laxatives are changing your microbiota, and they change the rate of your aging, or maybe even help to rejuvenate your body. So it's gut health and gut microbiome are absolutely critical for aging. Okay, it is accepted, yes, it is accepted that the gut microbiome can be healthy or unhealthy, and then the rate of your aging can be different. So it can be accelerated aging, or it can be even rejuvenation. Okay. So practical dietary consideration include gut transit time and protein digestion change with age. In infants, lactose functions as prebiotic, and transit times are very short. So sometimes it is 5-6 hours only. In adults, gut transit time typically ranges from 14 to 58 hours. People who follow strict carnivore diet, they report that gut transit time can be few days, up to a week. And this is not healthy at all. Okay. So in elderly people, protein digestion efficiency can decline substantially, leaving undigested protein, especially this. So some connective tissues and leaving undigested protein for these pathogens, proteolytic bacteria to convert protein into ammonia, hydrogen sulfide, and other potentially harmful compounds. And elevated pH, it is the pH which is the low pH is acidic, and so it has to be healthy pH should be acidic, but elevated pH and prolonged transit times can lead to dysbiosis and constipation. It is very common also. Again, if you on strict carnivore diet, it works perfect when you are young, but in old age, it has to be modified. So reducing gut pH to 6 and below favors beneficial bacteria like lactobacteria and the production of short-chain fatty acids. And these short-chain fatty acids, especially butyric acid, support gut health. And preclinical studies show protective metabolic effects or even of acidified water in mice. So if you drink lemon juice with water, even this can help, can shape your gut microbiome. Even this. So drinking lemon juice, why not? Why? It's easy and very healthy. Okay, so what we have, the colonic transit time, if it is increasing, then with age, then what we have, this number of some these pathogens is increasing and pH is increasing, so it is getting alkaline, the pH getting alkaline, and then you have this constipation and other problems. Again, this is, and then it is your aging is accelerating. Okay, so what is necessary to provide these prebiotics, and then beneficial bacteria will use these prebiotics as food, and then pH also will drop. It will be acidic, and acidic pH also stimulates peristalsis, moving of this feces through the gut. Okay, resistant starch type 2. It can be found in raw potato and cassava starches. It is about 60–70% of raw starch, raw potato and raw cassava starches as powder. It is not all, mind that it is not all. 30–40% can be digested in your small intestine. So also other fermentable fibers like inulin, chitin, lactulose. Lactulose is not fiber. It is actually the substance made from lactose, but it cannot be digested, and it is used as laxative. Chitin in mushrooms, for example, also work as the same as fermentable fibers. Inulin in fruits, in principle, can be used as the same way to improve health of the gut. But if you eat fruits, then you get also pectin. Pectin is considered as soluble fiber, but it is in fermentation of pectin, you produce methanol. Methanol is toxic, very, very toxic. So I would not recommend you to eat fruits. Inulin, I don't know. I didn't use it. Maybe people can use it. Lactulose, I cannot buy it, but it is very, very healthy, very, very helpful to improve your gut health. So this, among all this type of dietary fibers, resistant starch is the best, in my opinion. It is very affordable, very cheap. 1 kilo is $1 only in Taiwan. So it feeds butyrate producing microbes, and butyrate supports butyrate is butyric acid. It supports intestinal mucosa. Mucosa, it is a jelly-like layer on epithelium of the gut, protects the epithelium layer. It has anti-inflammatory properties, also reduce colon cancer risk. It also shortens gut transit time and modulates gut pH, so it is very, very, very healthy. So resistant starch type 2 can be integrated into low carbohydrate or ketogenic diet, also to carnivore diet, with minimal impact on blood glucose or ketosis, offering gut health benefits without compromising metabolic goals. Again, mind that a part of this raw potato starch can be digested in small intestine, so your glucose is getting a little bit slow up, but you can reduce this digestion of the starch in small intestine by combining it with mega doses of ascorbic acid. This is what I do. Yeah. So lactose can also act as prebiotic in some contents. Some people cannot digest lactose, so they can drink milk, and then it will produce it will work as laxative. Why not? Again, drinking 2 cups of milk per day, absolutely healthy for all people. So it is, again, if you have problems or drinking milk, these problems are coming from your gut health. If your gut not healthy, if you have so-called gut leaky syndrome, then you will have problem of drinking milk. But so if you slowly improve health of the gut, for example, with this resistant starch, you can drink milk later, no problem, absolutely no problem. I can live on milk. I consume a lot of milk because I am lactose tolerant people, so I can drink unlimited amount of milk. So and I consume a lot of milk, so it's very healthy, I believe. So typical recommended prebiotic doses are from 15 to 50 grams per day. It is recommended by official medical professionals, and but I use larger dose, like from 50, some days even 100 grams. It depends on yeah. Pardon. So and so, but don't take rapidly. I mean, don't instantly. So you need some gradual increase. Okay, otherwise it will cause gastrointestinal discomfort. Okay, so how is this gut health improving? So a proposed practical low-cost systems-based approach to extend health span, and so this includes lifestyle first. Diet, exercise, sleep, and avoidance of environment toxins from fruits and vegetables, rather than reliance on costly supplements and treatments. Again, it is very affordable. Microbial support, increase intake of fermentable fibers like resistant starch type 2. Chitin from mushrooms also work. And physiological monitoring, track simple physiological signs like blood pressure, resting heart rate, lung capacity and endurance, and as objective markers of and the feedback on interventions. Improving the elasticity of the skin and tissue is also vital for health and for longevity, but it is yet a matter of future research. I'm not much investing much, but what I use, I apply some substances to my scalp to grow hair. It works. So be cautious with experimental genetic reprogramming like Yamanaka factor in high risk and high cost procedures. It is very, very dangerous. Okay, so you can go to the source information. You see I rely not on opinion of medical professionals, but on scientific publications. Medical care and has nothing common with science, nothing. Medical care is business, okay? Mind that it is business, and then you cannot rely on the opinion of the medical professionals. Okay, thank you for watching this video. I share my personal opinion as a scientist on this, and this is, so I'm not supposed to give you advice, but I follow my findings, I follow, and it works for me. And it also worked for some of my clients. Okay, so also to support my channel, it is easy to press like and bell button, subscribe to my channel, share this video with your friends and family. Yeah, my hope to live forever, or at least a few hundred years, and I believe it is possible. So have a wonderful day. Ciao, ciao.